And we've got to stop our sodium coming in, okay? So we have to deal with our acetylcholine first. What's the enzyme that breaks down acetylcholine? Acetylcholine esterase. Right, acetylcholine esterase, ACE means an enzyme. And we said this enzyme simply cuts acetylcholine in half. It's gonna break it, right? So it can no longer function, right? Once that happens, right, it's gonna pull them off these receptors. Once that happens, sodium can't go in. So our cell will repolarize. We have that sodium potassium ATPase going, right? It's moving all that sodium out of the cell. So the inside of the cell will lose its positive charge because it's moving sodium out. Does that make sense to everybody? All right? So this signal will stop, right? Now, we need to move calcium back in the SR. We have low levels of calcium here. We always have high levels of calcium here. So we're moving against the gradient. Is everyone with me? What kind of things do you, what kind of transporter do you need to move something against a gradient? Active, right? So we're going to use ATP energy to move calcium back in, right? So we stop this, we stop the signal, right? We stopped our rush of sodium into the cell. We've stopped this electrical signal from traveling, right? Then we're going to take ATP and hydrolyze it, and we're going to start moving the calcium from the cytoplasm back into our SR. That make sense? So when we start pulling calcium out, what's going to happen to the calcium bound to a troponin? It'll release, right? It's going to release that calcium. As soon as troponin doesn't have calcium, what happens? Tropomyosin blocks it, right? And that stops the contraction. Is everyone okay with that? All right. Now we talked about rigor mortis, right? Rigor mortis, the key is when you are dead, you no longer make ATP. Everyone buying into that? When you're dead, you don't make ATP. So if we don't have ATP here, right, what happens to calcium? It's going to stay, right? We can't put it back in the SR, so it just stays. What happens to our thin and thick filaments then? Right, they can interact, right? We've exposed that site. Our thin and thick filament will now be able to interact with each other. What has to happen to the head of myosin to get it to release from a thin filament? You remember that? ATP, right? So our head's going to be bound, right? Head's going to be bound. To get it to come off, you have to have ATP. When you're dead, do you have ATP? No, so it will stay attached, right? That makes the muscle contract and stay contracted, right? Because you can't release it and you can't move calcium. So it stays in a contracted state, and that causes rigor mortis. Does that make sense to everybody?
So you can't release calcium?
You can't pump it back in. You can't pump it back in. So calcium will leave and you can't
Doesn't
pump it.
rigor mortis usually take a few hours or whatever?
To set.
Yeah.
Yeah, it will take a few hours to set, and then there's actually enzymes that come and break down. You'll have the lysosomes actually explode and will come in and tear this up. So it will actually go away over time too. That's correct. Okay, so you guys are pretty much okay with starting, stopping contractions? All right, so now what we need to do is talk a little bit about energy and kind of what contractions look like. So the way that we're going to look at a contraction is we're going to look at it if you give a single stimulus, right? One electrical signal. We're going to activate a neuron and then turn it off, okay? When that happens, right, here's our stimulus being given. And you'll note that there's this little delay before we get a contraction, right? Do you remember all the steps we went through to get this to happen? Right, we had to have release acetylcholine. Acetylcholine had to drift across and bind to receptors. Those receptors caused channels to open. Sodium had to flow inside. That had to happen all along the membrane. Then it had to get to our SR. SR then had to release calcium. Calcium then had to go bind troponin. Is everyone with me? Right? So why do you have this period called the latent period where nothing is happening to the muscle? Yeah, we had to go through all those steps, right? That takes time, okay? So that's what this latent period is. That latent period was the first step, then this happens, then that happens, then this happens, then the thin and thick filaments can interact, right? So everything that happens before those thin and thick filaments could interact, that's the latent period. Nothing's going on at the muscle. Then we get this contracting phase, and that's where we're developing tension, okay? So this is a look at a tension curve, right? We're looking at how much tension is being developed. Then we said, okay, we gave one stimulus, right? Then we went through and broke it all down, right? We took acetylcholine, we broke it in half, we stopped our electrical signal from traveling, we pumped back all the calcium inside, and that's causing the muscle to then relax, right? We're stopping the contraction, right? This is called a twitch. Anytime you give a signal, have the tension curve go up and then come down, that is a twitch. Your muscle twitch, okay? I'm going to skip these two. We're going to come back to them later and talk about this, okay? Now, the tension that we developed is due to the amount of cross bridges that are formed. So let me give you an example. I'll show you. The one that makes most sense to me, the example, is congestive heart failure, right? And that's when your heart's not pumping effectively, right? So what happens to the amount of blood if your heart's not contracting well? It's going to fill up. And if it fills up so much that it's going to stretch that sarcomere so that you don't have that many cross bridges forming, you're not going to be able to produce as much tension as if you have something that's not stretched as much. And you can form more cross bridges. Does that make sense to everybody? Now, you can have the muscle not stretched enough where the ends of the thin filaments are already so close together, you can't shorten them that much more, right? So that's what is going to affect the amount of tension that is developed. Here we don't have very many cross bridges forming. You're not going to get a lot of tension. Here, our thin filaments are already overlapping. We're not going to be able to contract them anymore because they're already together, okay? So you don't get a lot of tension. But if you get them stretched just the right amount, you get the most tension produced. Does that make sense to everybody? All right? So one thing that will affect tension is the number of cross bridges, okay? Other things that will affect the tension is the calcium ion concentration in the sarcoplasm. If someone is hypocalcemic, what's that going to do to the strength of the muscle? Hypo is too low. Will that affect muscle? Yeah, because you won't have the calcium to move the tropomyosin out of the way. So there will be a decrease in tension that way if you have too low levels of calcium. Does that make sense to everybody? Okay. Now, I said one of these little bumps was called what? Twitch, right? So now what we're going to do, instead of giving one stimulus, we're going to give repeated stimuli to the muscle. So we're going to fire that neuron over and over and over again, okay? So, if we take and deliver a stimulus, that's the green arrows, right? You can see the twitches that are being produced. Okay? If we give a signal so fast that our twitch doesn't have time to return all the way down to resting, right? See what happens to our tension, right? It's going to go up very high. If we give an electrical signal so fast that we don't see any individual elements, right? You're not seeing little peaks from the individual twitches, right? We produce the maximum amount of tension, okay? So we said cross bridge formation, the number of cross bridges that you have is important, and calcium amounts are important. Also, the frequency of the stimulus you give can strengthen or can increase the tension, okay? So, give you an example. This is simply contracting your muscle and keeping it contracted, all right? This right here is called incomplete tetanus. Okay, so we have a single twitch. Then we have, oops, this is incomplete tetanus. It is incomplete because we can still see individual elements, right? We know where our stimulus is coming in, right? And then we have complete tetanus. Right? We're giving an electrical signal so quickly that we lose all elements of individual responses. We're contracting our muscle and simply keeping it contracted. That is when you build up the most tension, right? Complete tetanus, all right? So the twitch has the least amount. Incomplete is going to be greater than the single twitch, less than the complete
So
tetanus.
is that complete tetanus like when it was a spasm? How like just tenses up like fully?
So it depends on what you mean by a spasm. You can have a muscle spasm start and
You
end.
know like when your calf like literally just like flexes for like an extended amount of time, and it hurts like.
Charlie horse.
No.
It's gonna, I'm not quite sure if your example is right, but it's going to be if you are contracting your muscle and keeping it contracted. So if you're doing like a tug of rope, and you sit there and contract that muscle and keep it contracted, as opposed to contracting, relaxing, contracting, you will have the greatest amount of force if it contracts and stays contracted.
Yeah, why I thought of this because like in wrestling, whenever we get like a muscle spasm like that, they tell us to drink milk because we'd be.
You're gonna rehydrate and everything. Yeah, that may be true. It depends on kind of your definition of spasm, where it's contracting and staying contracted and develop the most force. Okay? All right, so give me 3 ways to increase tension in a muscle. What do you want to do to cross bridges?
More.
Kind of an optimal number. Nice overlap, the thin filaments aren't too close together, but there's enough cross bridges to contract. What do you want to do to calcium? Add more. Optimal levels of calcium. What about the neural stimulus? High frequency, right? High frequency of neuronal stimuli to the muscle. Does that make sense to everybody? So, what we need to do now is talk about how the neurons are set up to talk to muscles, right? Each neuron will innervate a muscle fiber. Now think about that. You have a neuron innervating every muscle fiber in your body, okay? So we've got one motor neuron with all the muscle cells and muscle fibers it stimulates is called a motor unit, okay? So let me give you a picture of that. We've got one neuron, and these neurons can branch. And this motor neuron is innervating 3 muscle fibers. These 3 fibers are then a motor unit. They're a motor unit because you have one neuron innervating all 3 of these. Does that make sense? Right? You got one neuron, the motor fibers that it innervates are the motor unit. Okay? If we stimulate this neuron, all of these will contract. Okay? So we stimulate all muscle fibers in the motor unit. The muscle tension, right? If you're looking at an entire muscle, is controlled by the number of motor units contracting. Let me show you the picture here. All right? So here we've got a muscle, right? Now the motor units are in color, right? So one motor unit is all red. See the red muscle cells being innervated by this red neuron. That is a motor unit. Here we've got a blue motor unit, right? All the blue muscle fibers being innervated by the blue neuron. And then you got purple, right? Now, typically they fire asynchronously. You don't set all of them loose all at the same time, right? Here we've got the red one firing first, That one's gonna relax, we'll start the blue one firing. That one will relax, we'll start the purple one firing. To keep that tension level. Does that make sense? So we've got asynchronous firing, okay? Now, if we wanted to increase tension, the other thing we could do is we could recruit all the motor units all at the same time. So we can activate them all at the same time. If you have all of them contracting all at the same time, that will also increase tension. Does that make sense? Okay, but typically they fire asynchronously. All right, so usually fire asynchronously to maintain a contraction. Okay, you can recruit more to increase tension. So you can add more motor units. If you want to increase tension, you recruit more motor units into your contraction. Now, your hands, the motor units are very small, 3 to 4 muscle cells per neuron. In your legs, you have hundreds. Okay, tell me about motor control in your hands. You want anyone operating on your brain with your feet? Tell me about motor control with your hands. Precise, controlled movement. And that's because you have very few fibers per unit in your hands. You can control 2 or 3 muscle cells at a time, right? Very precise movement. Your legs, right, you're controlling hundreds of motor muscle fibers at the same time. That's very much a gross motor control. Does that make sense to everybody? So you've got fine motor control, right, when you have few fibers per unit, very precise control. That's typically hands, right, face, very few motor units per, or very few muscle fibers per motor unit. Okay, so we know that we've got a neuron and it's going to innervate muscle cells, and these muscle cells are all part of a motor unit, okay? The other thing that's important to know is that this unit is going to have the same characteristics, right? So this muscle fiber will behave the same way as this muscle fiber, which will behave the same way as this muscle fiber. Those purple muscle fibers will behave differently, okay? And so we're going to talk about, well, what are the characteristics that are making these all behave the same way? What do we look when we talk about it's going to act the same way? What are we talking about? One of the things is the way it gets energy, right? So this group will all get energy the same way. So when a muscle needs energy, you know it has to have ATP, right? The ATP binds to the myosin head. We're going to use that. We can also get energy from creatine phosphate. That is considered a storage form, okay? So when the muscle needs lots of energy, we can tap into creatine phosphate, which is a storage form. We can also make ATP from glucose, glycogen, fatty acids using aerobic metabolism. Right? So you remember how we talked about aerobic metabolism to make ATP? Was that producing a lot of ATP or a little ATP? A lot, right? So we would get a lot of ATP this way. What about anaerobic metabolism when we were using glycolysis? Little bit, right? Not very many ATP molecules per glucose. So these are the options that a muscle has when it's trying to get energy to contract, right? ATP, creatine phosphate, different forms of food, glucose, glycogen, fatty acids, and then it can switch over to aerobic metabolism. Okay, so myosin, where does it get its energy, right? Do you remember how we had ADP and inorganic phosphate actually on the head of myosin when it was resting, right? It can use that energy. It can use creatine phosphate. And I'm going to show you kind of what the advantages of creatine phosphate. You remember when we were starting with glucose and making ATP? Do you remember the number of steps we went through? We broke it in half, ran it through glycolysis, and we shoved it off into the mitochondria, ran it through the TCA cycle, pulled off all the little electrons, ran those through the electron transport chain, ran that through an ATP synthase, right, to make ATP. Okay? Thank goodness, creatine phosphate is much easier, right? You take creatine phosphate, and it reacts with ADP. What happens is this phosphate comes over here. What happens when you pull the phosphate off and add it to ADP? ATP, right? That's really easy. That is a one-step mechanism to generate ATP, right? Now, if you're at rest, right, and you want to store energy, you take ATP plus, right? We're going to take one of the phosphates off, move it over to make the storage form. one step. So you take and make ATP when that muscle is working very hard. When you're resting, you're going to make this, okay? When you're resting, you're gonna make it, right? So very, very straightforward, one-step process, either to make the storage form or tap into the storage form. We've got glucose, fatty acids, glycogen that we can use for aerobic oxidation, and then we can use anaerobic oxidation, or anaerobic metabolism, glycolysis, to produce ATP. And this is used when oxygen is low. Why do we use glycolysis when oxygen is low? Doesn't require oxygen, right? So when we have low levels of oxygen, we switch over to anaerobic metabolism. Lactic acid is produced. So this is a waste product of anaerobic metabolism. When your muscles get sore after you exercise a lot of it is due to the accumulation of lactic acid. Okay? So those are kind of our choices. So let's take a look at what's going on in a resting muscle, right? In a resting muscle, what we're doing is we're storing energy for later on, okay? So in a resting muscle, we're going to take fatty acids and oxygen. We are going to make ATP. That ATP we're going to slip into a storage format. We're going to react with creatine to make creatine phosphate. This is our energy storage, okay? Now with glucose, what we can do is we can store glucose for later, right? Muscle is one of two organs that makes glycogen, right? What's the other organ? Liver. So muscle and the liver make glycogen, right? So in a resting state, we're going to make glycogen. Glycogen is a storage form of glucose, which we may need later on. So we're going to make glycogen, or we're going to make creatine phosphate for in our resting muscle, right? We're going to store those supplies up for later on. When we move into moderate activity, our muscle's no longer resting. It's taking an ATP is binding to the myosin head, and we're cycling through the cross-bridge formation, right? We need to use ATP. So what we're going to do is we're going to take glycogen, right? What's glycogen a storage of? Glucose. We're going to take glucose, run it through glycolysis in the Krebs cycle to make ATP, right? So we're simply breaking down glycogen and making ATP. Muscles are going to use that ATP to contract. Okay. If we're at peak muscle activity, right? Peak muscle activity, we're exceeding the amount of ATP we can make with oxidative respiration, right? So we're going to break down glycogen, glucose. We've got no oxygen coming in, so we're switching over to aerobic metabolism. We're going to tap into our creatine phosphate, right, our storage form, and use that to make ATP as well. Okay? Does that make sense? So resting, we're making creatine phosphate, we're making glycogen. Moderate activity, glycogen. Peak activity, glycogen and creatine phosphate. Okay? Now, one way that we can continue to use aerobic metabolism is to have enough oxygen. So muscles actually have a way of storing oxygen, right? They use myoglobin, right? So myoglobin is like hemoglobin. What does hemoglobin do? Transports oxygen in the blood. Myoglobin is going to bind oxygen and keep it in the muscle, okay? It's going to bind oxygen and keep it in the muscle. So myoglobin will store oxygen in muscle. Okay. Now, this gives the muscle a particular color. Do you know what atom is found in hemoglobin? What metal ion is found in hemoglobin? Iron, right? Right? Iron is found in hemoglobin. Iron is found in myoglobin as well. Do you know what color this is? Red. So you guys do dark meat, white meat, turkey, right? This is giving you the dark feet because of the iron found on myoglobin. Okay? And so what is that telling you about what we're doing as far as how you make ATP? This type of muscle would make ATP using aerobic metabolism because we're storing oxygen. Okay? No myoglobin, right? You're going to have less oxygen. And this is your white meat. Where's your triplets go? Does that make sense to everybody? Okay. So we can keep an oxygen reserve. Okay. Now, I just wanted to kind of give you an idea of how this is affecting muscle performance. We're going to go into this in a lot greater detail. Okay? Here we have a muscle that is supported by ATP and creatine phosphate. It's going to use glycolysis primarily. It doesn't have that oxygen reserve, doesn't have a lot of mitochondria. It can produce a lot of tension very quickly, but then what happens to that tension? Drops off because that muscle is fatigued. It doesn't have enough ATP to keep that tension going. Does that make sense to everybody? It doesn't have it. It's going to blow through its reserves and then it's done. Okay? This muscle is supported by aerobic respiration. How many ATPs can you make from one glucose molecule using aerobic respiration? 36 to 38. Lots. How much by aerobic or anaerobic metabolism? 2. So that's why you're blowing through it really quickly, is you're only making 2, right? Here you're making 36, 38, depending on how you count. Here we're going to build up the tension more slowly. It's not as high, but look what we can do with it. We can keep it going much longer. Why can we keep this tension going much longer? More ATP. We're not blowing through it as quickly. Okay? All right. So just really quickly, I want to talk about 2 different types of contraction, and then we're going to talk about muscle types. Okay? 2 different types of muscle contraction: isotonic and isometric. Iso was the same. You remember the same? We talked about isotopes, isozymes, isomers. It was always same, right? The same. Isotonic solutions, same osmotic pressure inside out, same length. Okay? This is where a muscle is going to create tension, but it won't shorten. Okay, now if you think about that, if I try to pick up this chalkboard, which is bolted to the wall, right? I'm going to try and pick this up. My muscles, am I producing tension? Yeah. Are they shortening? No. What if I went up here and picked up a chair? All right? Muscles creating tension. Are my muscles shortening? Yeah, because I moved the chair, right? So that's the difference between these 2 types of muscles, right? Isotonic produces tension, the length shortens, so we're going to produce movement, right? That's me picking up a chair. Isometric, same length, right? We have equal length, tension's increasing, but there's going to be minimal shortening of muscle. The movement is opposed by an opposite force. The bolts holding the chalkboard to the wall are not letting me move it. I'm producing muscle tension, but I'm not producing muscle shortening because the chalkboard isn't going to move. Okay? So with muscle tone, you guys have muscle tone in your back, keeping you sitting upright. That is used to maintain posture. These isometric contractions are used to hold you in an upright position. You get asynchronous firing of fibers. Remember how I showed you we were contracting one motor unit, resting another one, contracting. So these asynchronous firing of fibers will help with muscle tone. Okay, so now what we're going to do is talk a little bit about different types of fibers. Okay, now key thing again, I'm going to tell you, I told you this before, I'm telling you it again, right? We have all of these red fibers, right? Innervated by one neuron. We said that's a motor unit, right? Those fibers are all going to behave the same way. They're going to have the same properties, right? So if this one has myoglobin, what can you tell me about this one? Myoglobin. If this one uses primarily aerobic respiration to produce ATP, what are you going to know about these 2? Aerobic respiration to produce, they produce same characteristics, okay? So that's key. If you know what's going on in one muscle fiber of a motor unit, you know what's going on in all the other fibers that are there, okay? And it's because of that neuron. That neuron is telling them how to act. One neuron, they're all gonna behave the same way. Okay, so there are 3 different fiber types in muscle. There is slow twitch oxidative fibers. Fast twitch A, these are oxidative fibers, and fast twitch B, which are glycolytic fibers. Okay, I'm going to focus on the top one and the bottom one. Okay, so I'm going to focus on the top one and the bottom one. Okay, slow twitch oxidative fibers use oxidative respiration. So how are they making ATP? Where are they going to make most of their ATP? What location in the cell? Mitochondria, right? Why do we know that they're using the mitochondria to produce ATP? Oxygen, right? You have to have oxygen to make ATP in the mitochondria, right? Because you're going to make, remember how you had oxygen, you had the hydrogens, electrons to make water at the end? Had to have that for those electrons to land on, all right? They're very fatigue resistant. Why does that make sense? If you're using oxidative respiration to make ATP, how many ATP are you going to be able to produce? A lot more, right? And so that makes them fatigue resistant. If you have lots of ATP to use, the muscle will not fatigue as quickly, right? Postural muscles, right? Things that you're using constantly. Your muscles in your back are used all day long, right? To keep you upright. So that's an example, all right? Fast twitch B muscles, these are glycolytic. Where is the ATP going to be made? Cytoplasm. Right? Because that's where glycolysis occurs. These produce very rapid contractions, right? Look at the name. Fast twitch. That's telling you how rapid the contractions are being produced, okay? These fatigue easily. Why do they fatigue easily? You don't have a lot of ATP and you're going to blow through it really quickly, right? Arm muscles, right? Fast twitch, okay? Now, what I want you to do is look at the characteristics that we can describe knowing those things, all right? So I'm going to put them up here and we'll talk about them. So we've got fast twitch, glycolytic, and our slow oxidative, all right? You remember our myosin ATPase, right? We talked about the head of the myosin, right? And we said, okay, well, it's going to bind ATP, it'll release the thin filament, right? Then our ATP will be hydrolyzed to ADP, an inorganic phosphate. And that cocked the head and got it in a high energy state and bound it to the thin filament. Then our ADP, an inorganic phosphate, came off and that head pivoted to produce the sliding, right? When that goes very, very quickly, you have a fast twitch fiber, right? Because it's going to bind to move the filament, bind to move the filament, bind to move the filament. It's fast. So what can you tell me about the speed of the myosin ATPase in a fast glycolytic fiber? It's fast, right? What about a slow oxidative fiber? It'll be slow, right? So how much ATP are you going to need if you're hydrolyzing ATP very, very quickly? A lot. So are you going to run out sooner or later? Sooner. So these are very, very susceptible to fatigue. Here, we're going to have a slower speed flowing through the ATP. We're going to be resistant to fatigue. Is this starting to make sense? Kind of putting the dots together? Okay, so fast twitch, right? Hydrolysis of ATP is fast. By myosin. Is fast. Here it's going to be slow. Okay? Speed. How quickly is myosin hydrolyzing ATP? ATP produced. This is a glycolytic fiber, so we are going to use glycolysis to produce most of our ATP. This is very inefficient, so we cannot produce much. Right? Here, we're going to use oxidative, which is going to be aerobic. This is tension and fatigue, right? Do you remember that graph that I showed you earlier? Right? Fast twitch fibers, how quickly do they produce tension? Very quickly. If you look at the amount of tension produced compared to a slow twitch, they produce more or less tension? More. Right? This is 100. See how it's going to 100? This one's below 50. So we've got lots of tension. Okay. Now fatigue. This will fatigue quickly, right? It develop tension quickly and then shut off. So this is going to fatigue quickly. This one is fatigue resistant. Okay? Now, this takes a little bit of critical thinking, right? Glycogen. Glycogen is a storage of what? Glucose. Okay? So, if we have one glucose molecule here, and we use it to make ATP, how many ATP molecules are we going to get? If we use one glucose molecule here to make ATP, how many ATP molecules will we produce? 36 to 38. Okay? Which will require more glycogen to produce the same amount of ATP? Glycogen here. If we wanted to produce ATP, 36 ATP, how much glucose in the glycogen would we need? 18 molecules, right? Right? Okay, so we're going to have lots of glucose here, right? Because we're only getting 2 ATP per glucose molecule. So we're going to have high amounts of glycogen. Does that make sense? And then less glycogen here because we don't need as much glucose because for each one we're getting 36 ATP molecules. Okay, so here we have lots of glycogen. Here we have less glycogen. Okay. Now, in our fast glycolytic fibers, where are we making most of our ATP? In the cytoplasm. So do we need lots of mitochondria? No. What about here, in our slow oxidative fibers? We need lots of mitochondria. Okay. What about myoglobin content? What does myoglobin store? Oxygen. So do we need lots of myoglobin content? Myoglobin content here? No, because we don't need oxygen for glycolysis. What about our slow oxidative fibers? Lots. So, if I have a motor unit, one of my muscle fibers, I've analyzed it, right? And it's got lots of mitochondria, it's got lots of myoglobin, right? It's got very little glycogen. What do I know about every other muscle fiber in that motor unit? Same thing, right? So if you find one slow oxidative fiber in a motor unit, you know that motor unit is slow oxidative, all of it, okay? What is controlling it? The neuron, the motor neuron that's innervating all of them, is making all of them behave that way. Does that make sense to everybody? Okay, so if we look at the fibers, right? Here is actually a picture of slow fibers. Here's a picture of fast fibers. If you look at the diameter of the fiber, which is larger? Fast fibers, right? The fast fibers. So if you think about people who do weightlifting, the big, heavy, right? So they pick up a weight and they put it down, right? What type of contraction is that? They're building tension slowly or rapidly? Fast, right? right? Tension goes away fast, right? So they are working with these fast fibers, which are very large, right? When you do those kind of repetitions over and over again, you build up the diameter of the fiber. You get hypertrophy, big muscles, right? Slow oxidative, right, are smaller in diameter, right? These are your marathon runners, right? Do your marathon runners ever look like weightlifters? No, right? They're not working on building up these fast twitch fibers, right? They're distance running, right? Slow twitch, long distances, repeated. Lots of myoglobin, lots of capillaries, feeding that muscle oxygen and nutrients, okay? Okay, so I just wanted to give you an example. Gastrocnemius and soleus, and you're gonna get this in lab on Thursday. Gastrocnemius are big frog muscles, back of the leg, right? So when you think about what frogs do, they leap, right? What type of movement is that? Fast twitch or slow twitch? Fast. They go up and you come down. You're done. That's a fast twitch, right? Now think about, so that's gastrocnemius. You see, high amount of tension, goes away quickly, right? Soleus, that's more of the ballerina types, standing on their toes, right? That's the same motion, right? This and this, same motion, right? Ballerinas can stand up on their toes for a really long period of time, right? You don't need as much tension to do that. You need time, right? So that is a slow twitch muscle. Is that right? It's predominantly slow twitch, okay? Now, the gastrocnemius and soleus, they contain a mix of fibers, right? It's not that just have one type, but the predominant type in the soleus is going to be slow twitch, and the predominant type in the gastrocnemius is going to be fast twitch, okay? All right, last bit, we talked about that you have 3 different types of muscle tissue, right? You have smooth, you have cardiac, and you have skeletal. So we said the smooth muscle, no striations, right? Tell me about the control of this muscle. Voluntary or involuntary? Involuntary. You can't make it contract if you want to. It's not under voluntary control. Cardiac muscle, right? We said this is actually striated, has the stripes, right? What was important about the membranes between the 2 cardiac muscle cells? Intercalated discs. What do they contain? What type of junctions? Gap junctions, holes, so that one cell can communicate with the other cell very quickly and they share electrical signals. Okay? Is this under voluntary or involuntary control? Involuntary. You can't control your heart muscle thinking. All right? This is way more information that you need to know. You do not need to know this, but I just put it here for your information. So like cardiac muscle has sarcomeres, that's what makes it striped. Skeletal muscle has sarcomeres. Smooth muscle doesn't really have organization of the fibers, so you can't see the stripes. So this is kind of a summary of that information for you. Will not be on the exam. You do not need to know it. It's for your information only. Okay? Just the last thing I wanted to talk to you about is different types of exercise that increase the different types of fiber forms. Okay? So if you go swimming or jogging for an hour, what type of fiber are you working on? Slow twitch. And what happens when you do that? What happens to your endurance if you do that type of training daily? It increases. It makes those fibers more fatigue resistant, right? More fatigue resistant. And what you're doing when you do that type of activity is you increase myoglobin. What does myoglobin store? Right? You have the oxygen, you can make ATP using oxidative phosphorylation, aerobic metabolism. And it will also increase capillaries in the muscle. What are the capillaries doing? This is providing blood. What's carried in the blood? Right? So you're delivering oxygen, you're delivering nutrients to the muscle. Okay. Then the opposite is if you go and do weightlifting. So you're gonna go and lift weights at the gym, right? You lift weights, you drop them, lift weights, drop them. What type of muscle are you working on? Fast twitch, right? What happens with that is that you will increase, fast, you increase myofibrils. What are the myofibrils?
Muscle fibers.
Muscle fibers, right? The thin and thick filaments, your myofibrils, thin and thick, right? So we're going to increase the number of those. What's that going to do to the size of the cell? Increase it, makes it bigger. So this is your hypertrophy. We're going to increase the diameter of the muscle fiber, right? Big muscles. So you're increasing the diameter of the muscle fibers, hypertrophy of the muscle. Does that make sense? Kind of what you know already. Here, go lift weights, you get big fat muscles. Here, you go running or swimming for an hour and a half every day. You're going to increase your endurance by increasing the amount of oxygen delivery and oxygen storage in the muscle. Okay? That is it. All right. So what I'm going to do is I've got a practice quiz for you. It's not real, it's practice. And we will go over this next time, so you have time to work on it. I'm going to go ahead and start the nervous system. So you're going to work on this at home, bring it in on Thursday, and we'll go over it on Thursday. Okay, so do that, bring it back in, and we'll go over it on Thursday. We'll go over it on Thursday. Okay. So we've covered muscle, and we know that muscle is controlled by motor neurons. Right? We know that muscle contraction is controlled by motor neurons. So what we're going to do now is switch over and talk about the nervous system and how neurons work to control different things. Okay? So, function of the nervous system, right? Function of the nervous system. It is going to be the mechanism that we use to maintain homeostasis quickly. So this is much, much more rapid. So this is rapid response. Next term, you will hear about the endocrine system, right? The endocrine system is responsible for maintaining homeostasis, but it does it slowly, okay? So this deals with speed, rapid means of maintaining homeostasis. And the idea with this, what the nervous system does is it's going to collect information. Sensory information comes into the nervous system, right? So it's going to sense changes in the body, changes in the environment. I'm hot, I'm cold, my hand is on a burning stove, right? Collecting that sensory information. It is then going to integrate that information. What should I do in response to being too hot? What should I do in response to being too cold? And it will produce a response that is then sent out, right? Either to muscles or to glands or to other neurons, but it will send out the response, okay? So best example, right, is a reflex response. This poor soul has put his hand on a tack, right? That sensory information will come in to the spinal cord. This actually doesn't even go to the brain. It's handled at the level of the spinal cord. Spinal cord is part of the nervous system. Information comes in, you get integration. My hand is on the tack, what should I do, right? And then you get motor information going out that causes you to lift your hand, right? And that motor information actually goes a couple of different places. Biceps, brachialis muscle will bring your hand up. What are you having to do to your triceps? relax that, right? So you have information going to both sides of the arm to produce that movement, all generated from the spinal cord, right? That's the best example of what the nervous system does. Sensory information comes in, integration, response to changes in the environment. I have stepped on a tack, okay? So that's what the function is, okay? And I don't think we've talked so much about germ layers, but the nervous system comes from the ectoderm. You have 3 germ layers in an embryo: endoderm, mesoderm, ectoderm. The ectoderm is what produces the nervous tissue, right? Ectoderm is a germ layer, produces nervous tissue. It will differentiate, the ectoderm will differentiate into nervous tissue. First it's called the neuroectoderm, right? And this differentiation occurs on the dorsal surface, the dorsal surface of the embryo, right? You remember where our spinal cord is located? You remember the dorsal cavity, right? What's housed in the dorsal cavity? Brain, spinal cord, right? So it's going to happen on the dorsal surface of the embryo. Chordates, right? At the very first day of class, we talked about chordates have a dorsal hollow nerve tube, or nerve cord. Sorry, I spelled that wrong, right? Remnants of that remain. You actually have hollow parts in the brain and hollow parts in the spinal cord filled with cerebrospinal fluid. So this is, you still have remnants of this hollow nerve tube. Part of the brain is hollow, part of the spinal cord is hollow. Okay. Now, you remember how we were divvying up so far, in the class, we've taken the skeletal system and we've divvied it up into the appendicular and the axial, right? We're going to take and divide the nervous system up. We're going to divide it 2 different ways. We're dividing it anatomically and functionally, right? Anatomically, it's really easy. Central nervous system and peripheral nervous system. Central nervous system is in the brain and the spinal cord, right? So if you are talking about part of the brain, cerebellum, you're in the central nervous system, right? Peripheral nervous system is everything else. Now, people always get this wrong, so I'm going to make sure that you guys hear it from me, right? Cranial nerves are nerves that come from the brain, right? So cranial nerves are part of the peripheral nervous system because they are not the brain. They are nerves coming from the brain. They originate, right? Spinal nerves are not part of the spinal cord. They are coming from the spinal cord. So they are part of the peripheral nervous system as well. Is everyone okay with that? What's part of the central nervous system? Brain and spinal cord. If it's not the brain and the spinal cord, it is going to be peripheral nervous system. Cranial nerves, spinal nerves, right? Not part of the central nervous system, part of peripheral nervous system. Okay, functionally, we can divide it. And this gets to be, this actually makes sense if you think about it a little bit, but people don't like it. It's a little bit more complicated, right? We're going to take and split the body into 2 parts, somatic and visceral. Visceral means internal organs. Heart, lungs, small intestine, large intestine, stomach, reproductive organs, right? That's visceral. Somatic is not internal organs. It's going to be the skin, the muscle, the bones, right? Non-internal organs. So we're going to split it that way, okay? Then, right, we said that what's the main function of the central nervous system, or the nervous system? What does it do? Response. Takes in what type of information? Stimulus. Sensory information is coming in. And then in response to that sensory information, sends out motor commands. Okay? Now, an afferent pathway is information being sent to the central nervous system. Afferent. Efferent, E is for exit. This is information leaving the central nervous system. So we've got a somatic nervous system. We've got the afferent pathway bringing sensory information in. And we've got the efferent, E is for exit, sending motor commands out. Okay? Now, where is the sensory information coming in? What did somatic mean? Muscle, skin, right? So we have that information coming from those locations. Where's the motor information going to go? Is it going to go to cardiac muscle? Skeletal muscle, right? The motor commands have to go to skeletal muscle. All right, so let's do this one. Sensory information, where's sensory information coming from? Heart, lungs, do we have enough oxygen in our blood? Is our heart beating too quickly? What's our blood pressure, right? So sensory, what kind of pathway is this going to be called? All right, then we have motor commands going out. Efferent, right? This is actually given its own special name. This is called the autonomic nervous system. Really important. Control of your heart, control of your lungs, control of your bladder, right? All very important. So we're going to spend a lot of time talking about the autonomic nervous system. That is visceral efferent pathways. Okay? Right? Those commands are sent to the cardiac muscle and smooth muscle, right? Internal organs. Does that make sense to everybody? You guys okay? Okay. Now, we've got two different kinds of cells in the nervous system. We've got neurons, and we're going to spend a lot of time talking about how neurons work. These are the guys that send and receive signals. So these are the communicators, right? We've got neuroglial cells. Neuroglia, glia means glue, right? So these are going to be cells that kind of glue things together. You don't have connective tissue in the brain, right? So we've got to come up with another way another way of supporting and protecting the neurons. Neuroglia are the cells that do that. You okay at catching this? All right. So we've got neuroglia are found in both the peripheral nervous system and central nervous system. Okay? Neuroglia that are in the central nervous system, where are you going to see those? Brain and spinal cord, right? Peripheral nervous system, not in the brain and not in the spinal cord. Okay? Can I erase? Okay. So, we've got peripheral nervous system. Okay? Peripheral nervous system is really easy, right? You've got, we're going to ignore those. You've got one cell type that's found in the peripheral nervous system, and these are called Schwann cells. These cells produce myelin. And we're going to talk about what that is. But it's basically kind of electrical insulation around wires. So anytime you go to get an electric cord, it is wrapped in some form of insulation, right? Plastic. Okay? That's what the myelin is. Okay? That is the only neuroglial cell in the peripheral nervous system. Central nervous system, we've got a lot more options, okay? Central nervous system, right? Do you remember I said there are hollow parts of the brain and spinal cord, right? Ependymal cells line those hollow parts. So ependymal cells are going to line the hollow parts, right? The hollow parts of the brain are filled with fluid. You guys heard of cerebrospinal fluid, like spinal taps, where they're accessing cerebrospinal fluid. Ependymal cells line the hollow parts, and they're going to assist in production of cerebrospinal fluid, right? So they're lining hollow parts, and they make cerebrospinal fluid. Okay? Then we've got microglia. Micro means small, so these are small blue cells, and these are phagocytic. Do you remember when we talked about phagocytosis? What is phagocytosis? Right, it's going to go wrap around and eat something solid, right? So microglia are going to go around and eat debris, damaged cells, things that should not be in the central nervous system, okay? Next, we have astrocytes. And astrocytes are really important. They will actually control what goes from the blood into a neuron, right? So you've heard of the blood-brain barrier, right? Blood-brain barrier. Astrocytes are part of that. They are preventing some things from getting into the brain and allowing other things. There are certain types of cancer that will actually go hide in the central nervous system. Why do they go hide in the central nervous system? What's the advantage? Certain chemicals, chemotherapy, cannot get into the brain to kill off the cancer cells, right? And so you have to go in and irradiate or actually apply that chemotherapeutic agent into the cerebrospinal area, right, to get it past that blood-brain barrier. Hard to treat infections in the central nervous system? Why? Blood-brain barrier. You can't get antibiotics across the blood-brain barrier, okay? So blood-brain barrier is going to protect the brain, and it keeps things out, okay? So this is a big what can get in, what can get out type of cell. Oligodendrocytes have the same function in the central nervous system as the Schwann cells in the peripheral nervous system. So the Schwann cells produced myelin, right? They surround the axons of neurons. Oligodendrocytes are also going to produce myelin, right? So these have similar functions. We'll talk about this producing myelin wrapping around axons, okay? So those are the cells that are not neurons, right? So we've got lots of different cells that are not neurons in the nervous system. So here's a picture of some of the cells, right? We've got our astrocyte here. What was the function of the astrocyte? Keeping things from crossing, right? So here's our blood vessel. Look at the feet of the astrocyte. You see that sitting on the capillary, right? And you see this other side is wrapped around our neuron, right? So here's a large neuron. Whatever crosses into the astrocyte can then go into the neuron. So this is making a barrier. Some things can get in, some things can't, right? Next we have our oligodendrocytes. Our oligodendrocytes produce these big processes that wrap around axons of a neuron. An axon is simply a really long extension. We said these are our phone lines, right? Our neurons are going to be communicators. So these are our big phone lines, and we're going to use an electrical impulse to travel down these phone lines. We're going to wrap those extensions in a process of the oligodendrocyte. And this process has what's called myelin. Now the idea with this is that this increases the rate of communication. So we're communicating with an electrical impulse, right, or electrical signal. So we're going to increase the rate of that electrical signal. Okay, we're increasing the rate of that electrical signal. So if I said you have a myelinated axon and an unmyelinated axon, which is going to be able to communicate more rapidly? The myelinated axon. It's going to be able to send its signals much more quickly. Okay? I think that's it. Okay, up here is our ependymal cells. We've got a hollow space here. Ependymal cells line the hollow space. We're going to produce cerebrospinal fluid. Okay, functions, right, are astrocytes, blood-brain barrier. Oligodendrocytes form myelin. Have you heard of multiple sclerosis? Okay, do you know some of the symptoms of multiple sclerosis? Difficulty contracting muscles, right? Right? So if we pull off oligodendrocytes off of the axons of neurons, right, what's going to happen to our way of communicating? We're going to slow it down. We're actually going to interfere with it. And that's going to produce multiple sclerosis. Okay? Microglia, small phagocytic cells, they go around, clear up debris, damage, right? Ependymal cells are going to line open spaces in the central nervous system. What do these produce? Cerebrospinal fluid.
So when the multiple sclerosis, when they're looking for lesions, are those damaged oligodendrocyte?
Right, and if you look, what's so bad about this is, can you see this oligodendrocyte is myelinating this, myelinating this? It can actually myelinate 100 different neurons. So if you kill this one, you affect 100 different neurons. Okay, we will stop here.